The Duke Superfund Community Engagement Corps held its annual Community Advisory Board meeting virtually on July 17, 2020. We were fortunate to have presentations from 2 Superfund trainees and 1 investigator from the Nicholas School of the Environment to share their work with our board. The researchers also participated in discussions on topics related to our Community Engagement Corps throughout the day.
So I am Paige Varner. I work on Superfund Project 5. I'm a 4th-year graduate student in Claudia Gunch's lab. And our lab focuses generally on using bacteria and fungi to clean up PAHs and sediment. But I particularly am looking at bacteria cleaning up PAHs using gene transfer. So thanks, Tess, for the intro to genes, because hopefully that'll help you understand my stuff a little bit. So I am looking at polycyclic aromatic hydrocarbons, which is the contaminant in our Superfund site, or I'll be calling them PAHs. Those are represented by 2 or more fused aromatic rings, as you see in this picture. They come from natural sources, as you see on the left, such as forest fires, volcanoes, or other natural processes. But a lot also come from anthropogenic sources or human-caused sources on the left, such as burning fossil fuels, chemical spills, smoking, and even grilling out. So those black marks you see on your meat in the summer after you grill out, those actually contain PAHs. Low levels, so don't be too scared, but they are really everywhere, which also makes it hard to remediate, in addition to they bind heavily to sediments. PAHs can cause certain health effects, such as cancer, birth defects, and other systemic effects that we don't fully know the extent of yet. So our Superfund site is in the Elizabeth River, where there is pH contamination from creosote. The Elizabeth River is coming off of the Chesapeake Bay here in Norfolk, Virginia, and there are multiple sites that are being looked at for pH contamination. One that has been previously physically remediated is called Atlantic Wood Industries. That has been found to have 800 ppm of PAHs, and just to put it into perspective, the EPA's report on the maximum contaminant level for particularly the most cancer-causing PAHs is about 0.004 ppm. So there are a lot of less harmful PAHs that are included in that 800 ppm count, but it still puts it into perspective that there are a lot of PAHs in this river. The site that we're looking at, and I'll be looking at, is called Republic Creosoting. So once again, this site is contaminated from creosote, which is this kind of oily substance you see here on the left. And it has been used as a wood preservative and all along the Elizabeth River, and it has leaked out into the river. So this site has 300 ppm of PAHs, which is still extremely large. And this has not undergone any remediation, which is why we are looking at this site. So I mentioned physical remediation methods, and 2 of the most common ones are capping and dredging. So capping is when you bury contaminated sediment by putting a sand or clay cap onto the sediment. And dredging is when the sand or the sediment is physically removed from the site and is either treated off-site or just put in a hazard waste site. Both of these are very common, but they're also expensive and environmentally intrusive. In addition, especially if you're in a high tidal area, there can be wearing of the cap and can actually leach contaminants over time. So we're looking at hopefully a more eco-friendly and inexpensive way to remediate these PAHs and other contaminants using bioremediation. So bioremediation uses bacteria, fungi, or plants to degrade the contaminants. I'm specifically looking at bacteria, so that's what I'll be talking about. And 2 common ways of promoting bioremediation are called biostimulation and bioaugmentation. So biostimulation is when you add nutrients such as nitrogen or phosphorus to the environment, and it stimulates the growth of these PAH-eating organisms so that hopefully they can produce non-toxic end products. However, when these bacteria that can do this function are not in the environment, we often have to add them to the environment using bioaugmentation. And then hopefully they will achieve the same goal of degrading the PAHs into non-toxic end products. So in general, the goals of Project 5 of Superfund are to maximize pH biodegradation and sediments in the Elizabeth River. Another lab member, Josh, who's in this meeting, he's using fungi and bacteria and trying to create a biofilm to do this type of remediation. And also we have some people looking at organic amendments, so adding certain organic matter to promote PAH biodegradation. But in general, I'm just looking at the first aim of the project. So my focus is a process called genetic bioaugmentation, which is a mouthful. But basically, it just means that genes are being added to the environment in order to remediate the contaminant. So this is done via plasmids. Plasmids right here are these circular elements of DNA that are separate from the chromosome you normally think of when you think of DNA, and kind of what Tess was talking about. So these plasmids usually are not essential, but they give the bacteria an extra advantage. So in our case, this is the ability to degrade PAHs, but also a famous plasmid function is antibiotic resistance. So this is actually how most antibiotic resistance spread, is through these plasmids. And this is done by plasmid transfer. So this is where the bacteria with the plasmid is able to replicate the plasmid into a new bacteria, so that both of these bacteria have this plasmid that gives them this special ability. So I'm taking advantage of this by putting in a bacteria, hopefully into the environment that has this plasmid, and hopefully we'll be able to transfer the plasmid with the ability to degrade PAHs into the natural bacteria in the environment. This is more helpful to us than just simply adding organisms to the environment that can degrade PAHs, because a lot of times these organisms will die off because they're not well adapted to the environment and will be outcompeted by the organisms that are naturally living there. So this transfers the genes that give the bacteria the ability to degrade PAHs into organisms that are already well adapted to the environment. And oftentimes these contaminated sites have toxic conditions for certain bacteria. So hopefully we'll be able to assimilate the genes through the environment for longer survival and longer term, and eventually degrade the contaminant into non-toxic end products. So the first goal that I am looking at in my project is to track plasmid transfer and PAH degradation within a synthetic community and lab. So a lot of these relationships are very unknown because it's hard to track plasmid transfer. And we really don't understand these topics at a basic level, which is why we're kind of starting with understanding them in lab with synthetic communities and not natural communities, so we can understand it a little better. So there are, like I said, it's hard to track. So there are only a few studies that have assessed plasmid transfer and PAH degradation, but they have suggested that maybe slower growing bacteria with these plasmids can degrade PAHs better, whereas faster growing bacteria with these plasmids can be able to transfer the plasmids better. So I'm aiming to kind of figure out if this is true for pH degrading plasmids. So I plan to mix a bacteria with a plasmid with different mixes of bacteria that are kind of slow and fast growing, and mixing them together to measure the amount of naphthalene that's degraded. And naphthalene is the simplest PAH, I think, that I showed on my first slide. But then I'm also going to measure the amount of plasmid transferred to hopefully kind of tease out which bacteria do this best. However, like I said, plasmid transfer is notoriously hard to track, so I'm developing a method to do this. So in order to do this, we're actually inserting fluorescent proteins into our bacteria so we can track the plasmid by color identification. So as you can see here on the left, I have a bacteria that's kind of blue on the outside. You can see the blue. This is because I've inserted a blue fluorescent protein into that bacteria. So the bacteria with the plasmids are going to be labeled blue, such as the one right here. Bacteria without plasmids will be green, and the plasmids themselves will be red. So we'll be able to track when the red plasmid goes into the green bacteria that doesn't have the plasmid. And this is an image from the paper that I'm getting the method from, where they did this with a microscope, but we'll be doing this in a little bit of a different method. So my final goal is to assess plasmid transfer and pH degradation with natural communities. So once we understand this at a basic level using the process described in lab, we can test these with native communities. So we'll be taking sediments once again from probably the Republic site on the Elizabeth River and adding different types of bacteria with plasmid based on what we know to see if we can promote higher pH degradation and plasmid transfer. So we'll once again be measuring pH degradation and plasmid transfer, which will be done by some DNA sequencing methods. So we want to find a balance between bacteria that transfer plasmids best and ones that degrade pHs best to get this long-term remediation. So this has applications with the techniques that I developed. We'll be able to better understand these processes. Understanding plasmid transfer has more applications than just pH degradation. So like I said, antibiotic resistance genes are often found on plasmid. So if we can understand this a little better, we can hopefully be able to limit plasmid transfer to limit the spread of antibiotic resistance.